Okay, welcome everyone to our May 27th edition of the Rutgers Cooperative Extension Ornamental IPM Program webinar. I'm Bill Errickson, Ag Agent from Monmouth County. We've also got Steve Rettke, our IPM Program Associate, and Tim Waller, our Ag Agent from Cumberland County. And our support staff, Erin and Quinn, who also helped put all this together. So just a disclaimer for the program. Again, we're going to be talking about materials throughout, so always follow the label. Make sure you're following the right materials for the state, if you're in New Jersey or in another state, and also know that neonics can only be applied in commercial agriculture by licensed applicators in New Jersey, so they don't apply for landscape setting, only for our nurseries and farms. But as you go through, whether you're managing a landscape or a nursery, a lot of the things we're going to be talking about will apply across the board. So we've got a good agenda for you today. Some of the insects and diseases that we're starting to see out in the fields right now, or things that are going to be coming up over the next 2 weeks to give you a good heads up. If you look at the bottom of the screen, we've got our current growing degree days. So southern New Jersey is a little bit farther ahead at about 640. Central New Jersey, 550, and northern New Jersey is at about 450. So that gives you the range throughout the state. And that box will stay up throughout the talk, and you'll be able to correlate that to the insects that we're talking about. And those growing degree days, again, they correlate with when the insects come out, and we try to really target their vulnerable life stages using those models. So for updates, always check the plant and pest advisory if you haven't signed up yet. This is a free service that we offer, and we send out regular pest updates as they happen. We also post the recordings of these videos and a downloadable PDF version of the slideshow that you can print out, or you can have on your computer. You can share it with your staff. And there's a lot of information in these webinars, so we just kind of highlight the main points throughout our talk today, but then we really intend the slides to be a supplement for you to be able to download. So definitely check that out once we finish the presentation today. So there's a lot of pest updates coming through on the plant and pest advisory. We also have been getting calls about spruce spider mites. We talked about those in their optimum control window a couple seminars ago. But we're starting to see the damage appear, so now we're starting to get calls. So check out the plant and pest advisory, and we'll send up some new updates on that as well. All right, so let's get into it. Starting off again first this week with one of our beneficial insects, and these are lacewings. So we're starting to see those out readily now, and these are really great beneficial. They feed on many different key pests. Aphids, caterpillars, lace bugs, beetle larvae, insect eggs, and even mites. And sometimes they're called aphid lions because they really can consume a lot of aphids. I'll show you some pictures. What we're looking at here, this is the larval stage here, and I'll show you some pictures of the adults and the eggs so you can know what to look for. We put these out there because you might see that in the field and think that that could be a pest, but it's actually beneficial. Okay, so we've got on the top left, that's a dusty lacewing adult. And then on the bottom center, that's a green lacewing adult. So that's the adult form. And then on the bottom left and on the right side, those are the larval stages. You can see on the bottom left, it's actually consuming an aphid there. So those will really go after aphids and consume many of those, helping with pest control. It's that larval stage that's really the most voracious. On the top center, you'll see those eggs too, right? You'll see them on a leaf or on a stem suspended by a silk. And so those are kind of unique, that if you see that and it seems curious in the field, that's what we're looking at. So you want to leave those and let them develop into lacewings that can help with some of this pest control. There was actually some research done a few years ago at Rutgers that looked at how effective green lacewings were for suppressing azalea lacebug populations in nurseries. And we always like to see the data behind some of these things, because if we're going to spend the money and the time to encourage beneficials or to purchase them commercially, we want to see how effective they are. And they found that green lacewings were really helpful in controlling azalea lacebugs. And there's many other pests they can control as well. Statistically, they were similar control to acephate, if you had between 5 and 10 green lacewings per plant. So we see a little bit of reduction in the percentage there, but if we're looking at the statistics, they were similar. So you get really good control using the green lacewing. So we can help to build up their populations in the field. And one thing we can do is to allow minor populations of the prey. So those pest insects, if the populations are small and we don't need to spray them, we can let them develop and be a food source for our predators, because if there's food, then the lacewings will show up. Especially if you're not going to sell those plants out of the nursery this year. You know, they can have a little bit of a tolerance for that food source. Also, avoiding broad spectrum insecticides, so they can affect many of our non-target beneficial predators. And we can look for lacewings in shaded areas. This is where we might find them and recognize that they're present. And then we can help to try to develop their populations by providing pollen and nectar sources. You can purchase these commercially, release them, and even mist foliage before they're released to help them to stick to the leaves. So the larvae are going to be more susceptible than adults to insecticide sprays, and those are also going to be our best predators. So remember, look for those larvae, and if you see them, try to foster their populations. Okay, so looking at our pests, these are some populations that we do not want to foster. This is a bronze birch borer, and the adults are active now. They're flying and looking for places to lay their eggs. So we're looking at the picture there. That is an adult female actually laying her eggs on a birch tree. So if we're scouting for these insects, they'll mostly be found on the sunny side of the trunks of the tree. That's a place that you can look for. And you want to look for these D-shaped exit holes. So it's when the adults are emerging and then they start their flight. And I'll show you some pictures of those. The adults, another thing we can tie this to with a plant phenology indicator is when the black locust trees are blooming, and those are in bloom or have bloomed in certain areas of the state. So they correspond well with that. Bronze birch borer will also, in addition to birch trees, they're also going to go after beech and hornbeam. So if you're growing those, you may want to scout those as well. And what happens is that it's the larva that really does a lot of damage. The adults are going to lay their eggs, and then the larva is going to hatch out, bore into the tree, and create those galleries mining through the tree. And what happens is then the tree is unable to conduct water and nutrients. We start to see a dieback from the top, almost looking like drought stress, because its conductive tissues have been damaged by this boring insect. So for controls, we want to try to take care of this before those larvae actually get into the tree and start boring. So there are some natural predators. There's also research that shows that birch trees that are not stressed are going to be more resistant. So especially drought stress on birch trees leads to higher populations and higher damage of bronze birch borer. There are some species of birch that are more resistant, especially our native river birch. So providing enough irrigation, right plant in the right location is going to help be a preventative measure. Also, if a tree is really damaged, we may want to remove that because it could be fostering increased levels of adults that could emerge. That could be a source of pests for the future. So for some of our controls, we can either use our contacts, which would be our pyrethroids, or the systemics that we have available for nursery commercial farm applications. Now, they're going to target different stages of the insect. So the pyrethroids, the contact materials, are going to target the newly hatched eggs. So, right, the adults are laying their eggs on the bark, eggs hatch, and then the larvae go to bore into the tree. So you want to get that material on the tree before the eggs hatch. So when the larva starts to try to bore in, it comes in contact with the pyrethroid, and it dies before it actually gets into the tree. That's going to be a good way to control them. Later, if the larva gets in, then we can use our systemics, which are going to get into the tree, and they can help to control the larval population if it's inside, but it's better to get that before it gets in there. It's also azadirachtin, which can be used as an injection as a biorational. And also want to mention there are US pest models specifically for this pest. And so this is included in our plant and pest advisory post, or you can set up a US pest model and it will show you, different life stages and which ones to target. So those are available. Another boring insect is the emerald ash borer. More of a problem from a landscape perspective and a major problem. We're losing a lot of our ash trees in New Jersey to the emerald ash borer. So we're going to see similar D-shaped exit holes as the adults are emerging. They're flying now, and they're starting to come out. Their activity is going to peak in midsummer. So the eggs are going to be laid by the adults on the bark. They'll hatch out in 7 to 10 days, and then the larvae are going to tunnel into the tree, similar to the bronze birch borer. So these are going to attack all of our Fraxinus species, all of our different ash trees that we have here. And we see a similar response from the tree, dying back from the tips back, like a drought stress response at first is what it looks like. And you see that D-shaped exit hole. If you're scouting, if you're out looking, you want to look for those holes and then look a little bit closer because it's a good indication that there may be adults or there were adults in that area at some point. So similar controls, encouraging plant health, encouraging some natural predators, removing infested trees that could be sources of future insect infestations. And then for ash trees, something really important is not to move ash wood. So for firewood, for example, moving it from one area to the next, that is kind of the biggest way that humans are transporting and spreading this to areas where it may not exist. Similar controls, looking at our contact materials as pyrethroids and the systemics. We also have emamectin benzoate as a drug injection that has been proven effective for emerald ash borer. But kind of the protocol for this is if you have really large, valuable ash trees that you want to preserve in a landscape, you want to treat them before they become infested. And it can be a process, it can be an expensive process, but if you really want to protect those trees, that's the way to do it. There are also models for this from US Pest, and we have that posted up on the Plant Pest Advisory so you can look at when to target the different stages of this insect. Okay, we're starting to catch some borers or clearing borers in our traps. We've been giving out free traps through the program. We've got them out at the research farms. We've been getting reports from folks. Thanks for sending those in if you have traps out, and it lets us know where they are and we can share that information. And so one of the insects that is starting to show up in the traps is the greater peach tree borer. And this affects all of our prunus species. So our flowering plums and flowering cherries are going to be affected. So really interesting here is that we have sexual dimorphism. And if you look at the picture, it means that the males and the females look very different. The males are on the left of this picture and the females on the right. And they look like two totally different insects. Now, the pheromone traps are only going to catch the males. So we're not luring any females into our nurseries or into our landscapes. We're only luring the males. And that's significant because the females lay eggs, and then the eggs turn into the larvae, which are the borers. So if we're trapping the males, we're not creating any more danger of bringing females into our trees to lay their eggs. So those are the two different versions of the sexes of the insect. Pretty interesting. So they're flying now, and the adults can lay up to 500 eggs during the season. These are going to continue to fly kind of most of the season. They're very active throughout the summer. So they've got a long flight period, long period that we want to try to scout for them. So we can see the picture up there of the trapped male right in the center. trapped clearwing borer male, that's kind of what it's going to look like when it shows up on the track. It's going to, you can see the differences there from the female that we had just observed. And then the borers, which we may not see, they're going to be inside the tree, causing that damage to the larval stage. You may see the gum exudate of the tree that we see on the right there. They kind of attack at the base of the trees, and we see this gum start to form from the tree as a result. Okay, reducing plant stress. Again, stressed trees are going to be more susceptible to our greater peach tree borer, and the pheromone traps are really going to be helpful for us in our monitoring to know when the adults show up. So we want to target the adults before they're able to lay their eggs and get the larvae into the tree. So that'll be our signal as to when they show up, if they do. When we're mulching, you want to avoid mulch volcanoes, right? Having mulch all the way up the base of the tree, because we could be hiding the area where the damage is being done. So if we're mulching, make sure it's not going up the trunk of the tree. Avoid damaging the tree, any physical damage to the bark, that can invite this insect as well. And if we have really bad trees, just remove them, cull them out, because they can be a source of future infestations. Okay, so we have some contact materials. If we can get them onto the bark during the adult flights, they could land, lay their eggs, and the larvae start to bore in, that will help. And then our systemics, if the larvae are present within the tree, that's when they're going to be most effective. We do have two biorational materials as a directin, and our spinosins are also going to be effective on this borer. And then the last insect I'm going to talk about is the black vine weevil. So this is an important one because it can affect a lot of different host plants, over 100 different species, our rhododendrons, our taxus, euonymus, Japanese hollies, hemlocks. So they can affect a lot of different plants. And then the adults are going to cause some damage to the foliage, but they lay their eggs, and then the larvae are going to feed on the roots, and that's where a lot of the damage can happen. So the adults are going to lay their eggs near the soil, and then 10 to 14 days later, the larvae hatch out and they start to feed on the roots. And this can be really problematic, and we may not see those larvae. So we want to scout for the adults now before they lay those eggs and before the larvae become a problem. So there's kind of a unique feeding that the adult does on the foliage. And if you see that picture up top there, it's kind of chewing up the edges of the leaf there. And it's not quite as dramatic as what like a native leafcutter bee would do, but they are chewing up that edge. And that can be a good way to monitor and to scout, to look for these adult populations. If you start to see that kind of damage, you think the adults are around. Then you can see the larvae feeding on the roots and what happens to the plants as an effect. So it can be a lot of damage that can occur underground. You may not know what's happening to the plant until we pull out the roots and see that they've been chewed up by these larvae. So looking for the adults first, they're active now. We can start to scout for them. So something that we can do also is place some wooden boards out on top of the mulch under the plants, and you can look under the boards in the middle of the day to see if the adults are there. They're going to go and hide in that shady, moisture rich area, and that can be a way that we can monitor for them. So contact materials, again, are pyrethroids. Once we see that the adults are present, we know we have them. Some of our contacts can help, or our systemics can help to get into the plant as well. So about 3 weeks after the detection of the first adult is a good time to start applying first foliar application, and then another application 3 weeks after that. We also have some biorational materials with a beneficial fungus that can help to control them as well. All right, I'm going to turn this over to Steve Rettke now to talk about some armored scales. Take it away, Steve.
Okay, hello everyone. Thanks for joining us again for this nursery greenhouse, not greenhouse, but I do a lot of greenhouse, but that's not what we're talking about today, but certainly insect disease problems. And we'll mention and go over some of these magnificent marvels of miniature malevolence that we have to deal with, we're forced to deal with. But I'm gonna be talking about armored scales again. And the ones I have listed there are 5, and they're pretty commonly seen in our nurseries. And if you look at the growing degree days, they're mostly going to be in the month of June and when they become active with their crawlers. Also, you notice that almost all of them, with one exception, are going to have multiple generations. And so we have our first generation beginning with 3 of the 5, and then we have the white prunicular scale that's now in its starting with its second generation coming up in maybe another couple weeks or so. And then we have the juniper scale, at least this far north, they only apparently have just the one generation of crawlers. And so this is the time of year when we start looking for these particular scales, and they're going to become active very soon if they're not already starting, which is what the euonymus scale is doing now. Okay. You're not seeing my photograph, are you, Bill? Okay. I'm just going to have to apparently say when next slide. All right. And so I just make a bit of a correction from last week. Even though there's more than 20 families of scales worldwide, there's only about a half dozen or so that we'll find in this part of the country, in New Jersey, for example. And there was maybe 1 or 2 families that I could be missing here. But if you notice, most of them are going to be phloem feeders, like I stated the previous time. And I made the incorrect statement that they're going to be all armored scales that feed in the cells, but actually there's another family of scales known as the oak pit scales that also feed in cells. But they're going to be pretty much only found on oak trees, and so it's not something you probably see a great deal in nurseries unless you have large field trees of oak growing. So, okay, next. I also wanted to make a little more effort and time to put into the controls of these scales because they're pretty much controlled all the same. And so this will be working for all the 5 that I'll be talking about today. And so if you're starting over toward the right, you notice I emphasize things like horticultural oils, which are really the ideal material to use against the crawlers. They work really well if the timing is correct and you get proper coverage. However, just remember, with the armored scales that overwinter as eggs, if you're doing a dormant type of a treatment, maybe even 3%, you're not going to get tremendous levels of efficacy with that scale with the overwintering eggs. And so they maybe perhaps get 30%, probably not a whole lot more. But so just remember that with the oils. And then we have some that are these insect growth regulators, which can be really useful, especially if you're trying to protect predators and parasitoids in particular. One is the pyriproxyfen, which I know best as Distance, although there are other brand names. And this is what is known as a juvenile hormone mimic. And what they will do is they will not allow the insect to properly go through its molting stages, especially when they get to the adult stage. And so they will not be able, with this material, it's a hormone mimic, it disrupts the natural functions of the scale, and they will not be able to become adults, and therefore they will not be able to lay eggs, which is important for control. It's not something with these IGRs that you go after with a large population. They're really best against crawlers also, but also when the populations have not built up to high levels. And there was a second one there, the bupropion, which I know best as Talus. And this mode of action is a chitin synthesis inhibitor. And what that does is the insects molt and they go through their various stages of development, they have to form a new exoskeleton. And chitin, which this particular hormone will inhibit, it's a chitin synthesis inhibitor, it will not allow chitin to be produced in sufficient quantities by the scale. And therefore, it won't be able to complete its molting stage and form the new exoskeleton over itself. And so that's also one that, just similar to the pyriprofen, that is not one you want to use against large populations, but work best against crawlers and then smaller populations. All right, there's a number of these contact materials that can be used. A cephate can be combined with oil, for example. A cephate is translaminar, and so that works particularly well against perhaps foliage type scales that are feeding on leaves. And then we do have the systemic insecticides at the bottom in the center. Materials like dinotepron and some of those others will work really well. Some university studies with dinotepron showing, maybe 90% or 95% control. And so those are still very useful. Okay. And then I wanted to mention, as far as some of these natural predators, which can be really critical for scales and trying to manage them. We have the green lacewings, which were talked about earlier. And we have lady beetles that would feed on scales, and then particularly these parasitoid wasps. And I do mention the fact that you want to try to prevent dusty conditions because they will negatively impact parasitoid wasps. Okay, next slide. And so this is showing a photograph of a lady beetle. That is actually one that's feeding on aphids. It's feeding on balsam twig aphids. But if it was the right species of lady beetles, it would feed on that scale there that's shown on the right, which is a cryptomeria scale on a Fraser fir. And it's indicating how there's like a big rough tear that what they will do is they will just simply rip apart the waxy cover and then go after that nice, soft, juicy material is what they're going after. Okay, next. And then the key one, I think, are the parasitoid and the parasitoid wasps. And the photograph on the left shows a female, and you've noticed that I have the red circle. That's her ovipositor, which she uses as an egg-laying organ. And I think most of you are familiar with parasitoid wasps and the fact that with armored scale, you'll see these exit holes. And usually with armored scale, there's one scale and only one exit hole. Whereas with soft scales, they may, because it's a larger scale, you may have multiple exit holes that could support more wasps to feed on the bigger scale that are the soft scales. But you see the exit holes, of course, as the larvae feeds, they're wasp larvae. The egg is laid through the wax and it hatches and then begins to consume the scale that's under the wax. And then as they complete their development, the wasp will become adults, and then they're trapped underneath the waxy scale with the dead female that's maybe totally consumed. And then they have to chew their way out, and they provide that exit hole. One thing that's kind of interesting to note, and a few decades ago at the University of Maryland, where they indicated that with the armored scales, what the female will do, and not always just simply lay an egg with her ovipositor, she will also puncture the scale in through the body and cause the scale to actually bleed. And you get the body contents of the scale begin to ooze up out of that exit hole. And then the scale adults can feed on that. And the males can't have this ability. And so the males are dependent upon the females if they want to feed as she punctures the scale and that ooze comes up out of the scale. Okay, next slide. All right, so we have another interesting study here done again at Maryland, where we have over a 4-week period. This is indicating, this is the chalcid wasp, which is a parasitoid wasp, and it indicates on the y-axis the number of these wasps that were trapped per day. And you notice the 4 different types of controls, well, 3 of them, with one of them being a control, which is going to be on each week on the far left side. And then you have the dark colored, which is a 2% oil that was applied. And then you have 2 broad spectrum materials that are the smaller ones that are shown. What that indicates, and I think if I remember right, the broad spectrums were Dursban, which is for pyrophos, which is really not used anymore so much in our industry, and then also the pyrethroids. And I think the one at the far right in each of the weeks, those were pyrethroids, and they last a long time. You see the negative impact it had on the number of these parasitoid wasps that were trapped per day. Some of them, they were showing like 5, 6, 7, 8 times reduction maybe like 85%, 95%, 90% reduction of these wasps. And it lasted over a number of weeks, as shown there with this graph. So next slide. And then this is also indicating interesting study again over multiple years now. And this is also comes out of the University of Maryland. The fact that if you're doing these broadcast cover treatments, a calendar treatment in a nursery, and this was actually done in a landscape, I believe. But this indicates with these three particular scale species, the euonymus, white pernicula, and white and pine needle scales, how it's interesting that if you do these cover sprays, which are basically blind sprays, maybe you get lucky and hit the crawlers some years and other years you don't. What you're doing is you're knocking out a lot of these parasitoid wasps and other predators as well. And what you're doing is actually encouraging scale populations to build. And so the y-axis there is showing the number of scales per branch as depending upon if you're been spraying these cover treatments over 4 years or more, that you're actually encouraging scale insects. Okay, next slide. And I showed this last time. It's just showing you that there's a lot of mimics out there. I don't want to spend any time on this, but this is a you might say euonymus scale. It's on a variegated euonymus. No, it's not. It's just bark callus tissue. All right, next one. And this, remember now, of course, if you're confused if it's got a scale or not, that any type of scale you remove the cover, or you're not necessarily even the scale itself, because only marmot scales can you really remove the cover by indicating that white waxy ring that's shown there on this particular branch. Okay, next slide. All right, so we talk about euonymus scale, and they're active now. If you still look at the growing degree days, they become active, at least central New Jersey now, for probably a good week or so. And you notice the yellow crawlers that are there on the upper left, and they're yellowish. And then some of the ones that are white are the males that were maybe the old males from the previous year. I think they're no longer viable. But then at the bottom, you've got, and usually males are going to be on the leaves, and then the females are going to be on the stem. There's exceptions, but the majority of that's the way it's going to be. And then on the stems at the bottom, you see the white and the gray. The gray are oyster shell shaped, and they're larger, multiple times larger, and they are females. And so you have, again, we talked about sexual dimorphism previously. The same thing is occurring here with these euonymus scales. And then in the center, you got, they will feed on Pachysandra, is showing the symptoms there, that yellow chlorotic material, chlorosis, where probably mostly males were feeding and causing those yellow spots to occur. And then you turn the leaf over, you see mostly males, they're white, and with a handful of females that are there as well. And then on the upper right there, you've got mostly white males on the leaves. Okay, next one. And a good close-up, by the way, of the sexual dimorphism with these euonymus scales. You can see the oyster shell-shaped female considerably larger, like 4, 5, 6 times larger than the elongated white male that's very narrow. And as you see the host plants there, when you look at, and this is just generally when you're monitoring, it's key to know, well, what plants are you looking at? And if, for example, if you're a monitor, like what I do in nurseries, I look around and look at plants. I see what the plants are. And immediately things come to mind. What is the common key pests that are going to be on these host plants? And if it was euonymus scale that was there, and at that time of year with the growing degree days, if you know that as well, then you begin to look at the variegated evergreen euonymus, or the Japanese Pachysandra, or even boxwood or bittersweet, Allegheny spurge. They are all potential hosts. And so that's kind of a 3-step process. You know the plant, you know the pest that they're key, what they're going to probably have a tendency to be infested by, and then you need the time of year. And so those 3 kind of really go hand in hand to help simplify your monitoring process a great deal. All right. And so pretty much talked about everything that's shown there. And so we have another one. We finished up with euonymus. This is Cryptomeria scale. And is this Cryptomeria scale was my question? And it's on a pine, and Cryptomeria will feed on pine. Well, the answer is no. This is actually a bristlecone pine, and that has a tendency to ooze out, sap from its needles, and that sap dries and becomes white in color. And so it could be mistaken as a scale, but of course it's not. Okay, next. And they're showing you the symptoms of Cryptomeria scale. on a Fraser fir needles. That's the top of the needles. You see that chlorotic mottling type of a look. And if you see that, especially if you're going to see it more often than not, if the population hasn't gotten too severe, it's going to be at the base of the plant and maybe toward the inner needles. Then you turn the branch over, that little twig over with the needles, you see all the scales. These are the signs. And so you have the symptoms on the left and you have the signs on the right, which is the actual causal agent. Okay. And so this is a fairly mature population because as these scales develop, they become more opaque, whereas earlier in their developmental stages, they are mostly almost clear. Okay, next slide. And just to reiterate that, look at that photograph there. That is a little bit younger cryptomeria scale, and you can see actually through it. It's almost a transparent cover. And they really are most susceptible with our Christmas tree farms. And I do see cryptomeria scale on a routine basis when I monitor Christmas tree farms, but they will feed on other conifers as well, like hemlocks, pines, and firs. And as I indicated there, emphasize the beware of the scale hiding beneath a transparent cover. And so that's something that you have to be aware of, and maybe look closer. You don't, perhaps you turn the leaf over, you don't even, well, they're not even there, but yes, they are. You just have to look more closely. They kind of look like a fried egg, and they're very flat in appearance. And their crawler periods can last a good 4 weeks or more, and they do have 2 generations that's shown there at the top. Okay. All right, let's go to the next one. I've got another one. It's the white paniculus scale. Is that a white paniculus scale? Well, maybe. What do you think? No, but no, they're dried seed oils. And what happened here? I believe this was bittercress. It was about this time of year when it's a winter annual, by the way. It's a broadleaf evergreen or broadleaf annual plant, and they're shedding their seeds and they forcibly eject them up into the air. And this was a plant leaf that apparently had some of the seeds land on them. Maybe the seeds, the late leaves were wet at the time and they kind of stuck for a while. And then over maybe a period of time, the seeds began to ooze out some oils. And then they were clear initially, but then they turned kind of a white color. But those are seed oils that dried up. Okay, next. And so here you got your white panicular scale, what they really look like. And I tell you, I've seen a lot of them over the years in nurseries, and particular as far as container plants. They're going to be at the base of things like blue hollies. It was almost inevitable. You look through a group of blue holly container plants, and there's going to be at least a few of them that have these white panicular scales right at the base. And the males and females are both white, but the males are going to be kind of have a waxy look to them, filament type of material that they have around their bodies. They're very narrow, whereas the females in this case are going to be circular in color and rather, but also white in color. Remember that with armored scales, all male scales are going to be elongated or elliptical in shape, whereas it depends upon the species of scales. Some female armored scales will be circular, or they could also be elongated as well. And so you can really build up these armored scales with the males actually obscuring the females completely. Okay, next one. All right, so as far as what we're looking for, you can see the host plants again, primarily blue hollies. It's really become prolific in nursery containers today. I remember in the landscape when I was there in the 80s working as a landscape industry, it was commonly found on flowering cherries, mostly on the lower scaffold branches, just caked, just a thick coating of these white panicular scales. And oftentimes what people would do is just, if it was within reach and was practical, they could just scrape the scales off and be easier to control if it was easy to reach them. All right. All right, so I think I've pretty much covered all this. Let's go to the next one. All right, what about this? Are these juniper scales? Well, these are rhododendron leaves. I don't think they're juniper scales, and so the answer is no, but these are actually artillery fungus spores. And if you're familiar with artillery fungus that decompose or mulch, wood mulch, they're natural wood decomposers, and they shoot their spores up in the air forcibly, and they're phototropic, and so they'll go into light colored surfaces, and sometimes they'll go up on white colored siding, vinyl siding, for example, they can shoot their spores, 25 feet or more up into the air, and they're relatively large. And you might mistake them. What is that? Is that a scale? But no, it's not. You remove that. It's not easy to remove, but you remove that with your fingernail. You probably can get it without too much trouble. It's not going to leave a white ring. So no, those are not scales. All right, next. All right, so these are the juniper scales. You can see them there. Again, with the parasitoid wasp exit holes, we've talked about enough. We know that we can test for scale viability by, you know, using a sharp pen or just using your thumb and forefingers and see if you get much in the way of scraping them and get a lot of body fluid. You know that their scales are probably still viable. There's the actual scale in the upper center. And then also to kind of indicate there that white outer ring that's very much present. And both the upper center photograph, you can see that white outline of the ring, as well as the lower one, which I think it's the same photograph, it's just rotated. And from a further distance, you can see that white ring as well. Okay, and there's the close-up of the female scale. All right, so I think I pretty much keep talking about the same thing as far as these scales and what they look like. You can see that they really only have even though they indicate a dormant period, that's not really a crawler period. You have the crawlers of just one generation this far north, and you have junipers and false cypress and Leland cypress and arborvitaes. You'll find them on there. Okay, and so what else do we need to talk about here? Just the one generation, as I've already stated. And they do hatch. They're probably not quite ready to hatch yet in central New Jersey. Getting really close to hatching down in South Jersey, another week or so, and then they can continue into July. And so this is another one that these armored scales. Remember, when they're crawlers, crawlers can be active over 2-3 weeks, and sometimes 4 weeks over a long period of time. All right, so next one. And what about this one? Are these the Japanese maple scales? Well, no, these are katydid eggs. All right, that would be kind of funny to mistake this as a scale, but these are actually katydid eggs. Okay, next. And this is what they look like, Japanese maple scales. They're on the left. You've got a twice stabbed lady beetle predator, very voracious. Can't really see the two red colored spots on the side of the beetle there, but it's a type of lady beetle that specializes feeding on scales. And then you can see how the scales can just be infestation, just encrusted like barnacles on a rock. And that's a big difference. I mentioned this the previous time. Armored scales, once they're present, they can last for multiple years and just keep building up. And that's when they eventually can actually kill trees, you know. And then what's really kind of critical about Japanese maple scales, if you're not familiar with them, because it's the only scale that really is elongated with both sexes. They're elongated and they're going to be found only on the bark, and they're white in color. You turn that scale over, you see that purple colored blob, and that is considered to be diagnostic. You see that, and you turn, that's a female, by the way. If you see that purple coloration of the female, as well as the crawlers are also going to be kind of a purple coloration. Okay, next. And finally here you got, again, that close up there of the scale where you remove that waxy coating that they have. You see that dark purple coloration, and it's really quite diagnostic. This is a scale that has just the 2 generations. You can see all the host range is pretty extensive. You got maples and firethorns and serviceberries and lilacs, cotoneasters and prunus. And so it has many that they can feed on. And it extends, crawler periods is really long, and they have 2 generations. And so this is one that you can start having, say in New Jersey here, central New Jersey, probably maybe the second week of June, we'll start reaching the growing degree days. And then they can be present all the way through September. That's a crawler period that's really quite extensive. All right, so I'm gone over, didn't do a good job. I'm over by 20 minutes. And so we're now magically, Tim Waller will appear, I guess. I don't know if he's going to have his photograph or not, but I'll bring it over to Tim Waller as next.
All right, well, hey everyone. We're going to talk about one more pest insect, and I've been sending plant and pest advisory posts out about this for a number of years now. It's one of the pests that got me into utilizing growing degree days and kind of understanding the nuances with them. So we're talking about a pest that can be a nuisance to a full-blown infestation and varying amounts of control practices that are implemented. In a lot of ways, it's kind of the beginning of the summer season here in southern New Jersey and all the pests that are going to start coming in. So we're talking about bagworms. In the image here, you can see, if you look at that little arrow, you see that tiny little bump right there. That's almost too big for us to get into contact material pesticide control of this particular pest. So what that means is we really need to keep an eye on when these juveniles are being released, and we're going to talk about that in a second. But really, timing when it comes to controlling bagworms is incredibly important. Just to kind of put things in perspective, a single bagworm can have 1,000 eggs that are released from that bag. So if we miss a few here and there, it can lead to a very large population in the next year because they are one generation per year. So it does list host plants here, but I've seen bagworms make bags out of all kinds of things and look like they're camouflaged. Whether that is, most of the time we attribute this to our conifers, but I've also seen them on deciduous plants as well. So what I'm talking about when it comes to understanding when we need to target bagworms is we're targeting them right after this period known as ballooning. So if we look at the image, the actual image, they don't ride around on hot air balloons. That's a Joe Boggs picture, by the way, or both of these are. But we can see that there's silk formed here. And basically what happens is as the eggs begin to hatch, they have a projection of silk. They come out of the bag and they get drifted around in air currents. This is why we can do an absolutely immaculate level of control in one season and then have bagworms show up in another season. If you are having issues with bagworms, make sure you are checking whether you've got windbreaks or you've got really large plantings of arborvitae, something that you're using as kind of a blind for your nursery operation. Make sure you're also checking them for bagworms. So, oh, there we go. So with bagworms, like I said, we really need to, well, we'll talk about this slide first then. When it comes to managing bagworms, if you have bagworms that are at this stage, this is an excellent picture taken by Steve Rettke, by the way. If you have bagworms that are at this stage, when it comes to using contact materials, we've pretty much missed the boat on that. These pests are extremely tough in the first place, and they have a protective coating, so it means they have to be moving around in that contact material. Something that's very important when it comes to bagworms is lethal doses. The rate to give them a lethal dose of a particular compound. There's been some research that instead of dying, once caterpillars are at this stage, they'll go into early pupation, meaning that they will be able to move on into the next season because they're already in pupation. Can we go back one slide? And so, like I said before, it can be a nuisance issue. A lot of times you'll see them higher up in the canopies. That being said, I've seen them all over the place. I've seen them high up in canopies. I've seen them in weeds at the bottom of plants. They can defoliate. But the central image here, and if you haven't done this before, it's definitely worth doing, is looking through bags. You very possibly could miss a bag that was made 2 or 3 seasons ago and you may be monitoring that particular location trying to see if you're going to have caterpillars emerging from it. There's a good chance that it won't. So you need to break those open, see if you have eggs in there, and give it a good squish because it'll give you an idea of whether it's viable. One of the things we don't talk a lot about with bagworms, and this is going to be in larger plants, is their ability to girdle and cause what looks like flagging or tip death. And that's something I've seen in a number of larger trees that have been afflicted with bagworms. All right, so there are a lot of materials. If we catch them when they're young, they're very easy to manage. We've got our very broad materials that we can use, the carbamates, pyrethroids. other materials where if we're targeting them when they're at a much smaller phase, that can be controlled very easily. We do have a number of systemic insecticides, the diamides and combination products thereof, that can be effective as the larvae are moving, or if we're using them in a translaminar kind of fashion. One of the materials, because we are talking about a caterpillar, BT, comes up, and that's something that can be quite effective, as well as spinosad or the spinosins. The other thing I just want to mention here, though, is that once we get past that really small phase, mechanical hand removal, I know it is something that is going to cost a lot of human hours and a lot of money because of that, but that is a really great way to get rid of them. Make sure you aren't just pulling them off and dropping them onto the ground. You're removing them or destroying them in some kind of way. All right, so we're going to talk about a couple plant diseases. And I'm going to try to get through this quick, and these resources will be available to y'all later. So, Voluatella blight. We work with boxwoods. Boxwood blight always comes to mind, and Voluatella blight is also very important, and they're often confused with each other. The reason that I put it in this particular visual here is that Voluatella, not only does it get on boxwood, it gets on sweetbox, and it also gets on Pachysandra. And I've seen this occur in nursery setting and landscape setting for all of these different species. This is definitely the warm weather pathogen when we're talking about boxwood diseases. Boxwood blight is more the shoulder, cooler seasons. Voluatella is much more the warmer period of the year, high humidity, i.e. our early summers into our summer here in New Jersey. This is the full description of this is Voluatella foliar blight and cankers. It'll attack the foliage and it'll also create cankers across the different hosts that it's attacking. On boxwood, a lot of the times you'll see these straw-colored leaves that are still stuck onto the plant. That's a big differentiator between boxwood blight, where the leaves are cast from that plant. On Pachysandra, you've got these large expanding regions of necrosis, of symptoms, and it's pretty obvious that you're dealing with a fungal infection. So just a couple visuals of Voluatella blight. We're seeing that expanding necrotic region. We're seeing the canker in the other corner there. In a landscape setting or in a large cutting system, there's absolutely the opportunity for this massive explosion of this fungal pathogen that can lead to a lot of death very quickly. It's also very easily moved around by humans when we're trying to propagate or to prune materials. And then we've got Voluatella blight of boxwood. So what it's going to look like most of the time, it's going to look like this region of straw-colored leaves, a little bit of casting. These leaves could look very similar to winter damage. However, in winter damage, you'll see regions that are beginning to grow again. You'll get regrowth within those straw-colored leaves. Like I said, it's not just a foliar issue. It also causes a canker, and we're going to talk about the next slide here. Under laboratory conditions or very hot and humid conditions, i.e. our summers in very crowded greenhouses or hoop houses, we can absolutely have the formation of what looks like these orange salmon colored, they're called sporodokia. They're basically the spores on the bottoms of our leaf surfaces. This is again different than boxwood blight, where we will see most of our symptom development on the top side of leaves before they're cast from the plant. So Voluatella blight, there are a number of materials available for it. Mancozeb comes up very often as a material that has really good impact for dollar cost against this particular pathogen. However, it's got a really long REI. Chlorothalonil is a great cover material as well. Coppers, there are some premixed materials that are going to be effective on this as well. And then we've got some of our other generalist systemic materials like T-methyl and the conazoles. I just want to point out that, and this is a conversation I have with many people quite often, is that yes, there are some materials that are going to have a 24-hour reentry. And I understand that's not ideal in most scenarios. However, this and another example I'm about to discuss, they should at least be considered if the management and the movement of people around that area can be modified for that time frame. Understanding it's not an option in all cases, but these are some really profound tools that we can utilize. The last thing I want to mention about Volatility is that sanitation is critical. If we've got cankers, we've got blighted materials, we need to get that out of the system, prune that away. However, we need to make sure we are pruning deep enough and we're not cutting through those regions. If we do, all we're doing is spreading disease, and you need to make sure that you're cleaning and sanitizing whatever pruning implements that you're utilizing. And just a quick plug here of giving the visual symptom, or the signs rather, of the different diseases that are attacking boxwoods. Volatility, if you incubate plant material and you've got salmon colored spores, and they look like kind of little dots, really good chance that you're dealing with Volatility, versus very, very fluffy, bright white mycelium. There's a very good chance that you're dealing with boxwood blight in that scenario. So sticking with the trend of knowing the difference between these, we've got powdery mildew and we've got downy mildew. Both of these are caused by many different species, but the key differentiator here is that powdery mildew is caused by a fungus and downy mildew is caused by an oomycete. The oomycete group also includes a lot of root diseases like Phytophthora, Pythium, and Phytopythiums. With powdery mildew, we're going to see that the obvious powdery mildew symptom that we've all seen before, but what's kind of cool about that is if you really look at it at microscopic level, that white material is actually the fungus, and we are looking at its asexual reproductive phase. If we kind of keep going clockwise here, where we've got the leaf with the tiny little black dots on the bottom, this is much later in the season, and what those little dots are, in plant pathology, we collectively refer to them as Pac-Men, but they're known as chasmathesia. And these are sexual phases that will be released early in the next season to reinitiate an infection. The last one in the corner there, that's damage to boxwood. Boxwood does not always show a white appearance. And you may also see foliage that has been damaged much earlier, where we're starting to see this kind of discoloration, crinkly appearance. So keep an eye out for that as well. So downy mildew is caused by many different species. It gets in a lot of our annuals as well as a lot of our food crops. It's very common in the greenhouse situation. And it's on many different hosts. Kind of an indicator of this is if you incubate this material, there's a very good chance you're going to see sporangia, or what looks like that image, that microscopic image there, protruding from the bottom of the leaf surface. On the tops of foliage, oftentimes we're going to see yellow. In some cases, we're going to see purple, and a generalized discoloration of those tissues. So there's some key differences because powdery mildew is a fungus, downy mildew is an oomycete. We're going to have some differences in the materials. So with powdery mildew, the DMIs are really effective. So this myclobutanil is one of those times where it may be worth using a material with a longer REI. There's a lot of research that this is a really good material, but it does have that caveat, understanding that can be really a big deal. That should be a 12-hour there. I apologize. Some other premix materials that are great options as well, and they are building us into this concept of pesticide rotation because it is very, very important when it comes to many of these fungal pathogens. We've got a lot of other materials here, and then this material here, which is regalia, is utilized in many cases as well, and that mixed into combinations or into pesticide programs can be quite effective as well. So with downy mildews, this from protectant, local translaminar, all the way down through that list, I pulled that over minus a few things from the Phytophthora and Pythium list. It's because those materials are made to attack oomycetes. But you will notice that there is some level of control from many of the premix materials that are out there, or at least labeled for it, as well as some of the more broad use materials as well. All right, so I was gonna go into a big thing about Cercospora leaf spot versus bacterial leaf spot, but we're getting kind of close on time here. But so Cercospora leaf spot, we're gonna talk more about this as we move throughout the season. But what's really important about this is trying to get a good diagnosis. Are we dealing with a fungus or are we dealing with a bacteria? In the bottom corner there, this is what's known as bacterial streaming. And we had a lesion that this person didn't know if it was fungal or bacterial. They cut through that lesion, placed it onto a slide with water, and they were able to see bacteria streaming out of that tissue. This is the indicator that you are dealing with a bacteria. With many fungal diseases, we're looking for a necrosis that is irregular in shape, meaning it's not like little octagon type shapes most of the time that are kind of coalescing into each other and destroying the tissues. When we incubate them, we will also get spores being formed. I included this particular list of information. Coming out of Tennessee State, it's been trialed, it's been evaluated, and these tank mix or pre-mix materials have been quite effective in managing Cercospora leaf spot in hydrangea. So this is a plug for the diagnostic lab. Many of the things that we've talked about, we need to know what we're actually up against. If we don't know if it's a bacteria versus a fungus versus an oomycete, we are going to make the wrong call in the materials that we're using. All right, so trying to keep us on time here, I just want to say thank you very much for attending. So these are the pests that we're going to go over next time. And what I want to say to the group here is these lists that we've been working on are the things that we deal with on an annual basis, roughly around these time points, or they correlate to growing degree days, and we're trying to give you a heads up of what might be coming. That being said, would absolutely appreciate if folks wanted us to talk about something that they know is going to be a problem that's coming up, or if they had ideas for specific topics that they wanted us to take a bit more of a deep dive into. That's something that we always appreciate, and we're trying to do this in a way that is the most helpful for you, our growers. Another thing that we really appreciate is calling one of us and letting us know if you're seeing a huge population of X pest or this scale has just emerged. And those of you participating in the trapping, please keep us informed and let us know what's coming up so we can make the best use of everyone's time. Another thing that we do appreciate too is if you're getting really good control with some combination of materials, rotations, etc., and you're willing to share that information, that can also better inform the materials that we're able to provide for everyone. So with that, I just want to say thank you very much, and we are going to quit recording and then see if anyone has any questions.